I'd like to review some simple genetics problems because understanding how a simple genetics problem works is really important to understanding the concept of inbreeding. And so let's say we have a genetic disease that is recessive, and let's say that genetic disease results in the death of a baby. Okay. Well, if the disease is recessive, being normal would be dominant, and the disease would be indicated by a lowercase n, which means it's recessive. So let's say you cross two individuals that are phenotypically normal but carry the disease. Well, if they're phenotypically normal but they carry the allele, that means you're crossing two heterozygotes. And so here we're showing you a cross where we're crossing two heterozygotes that are carrying the recessive allele for the genetic disease. Let's say we have the male on the left. That male has the potential of producing two types of sperm. And in fact, 50% of the sperm will contain the dominant allele, and 50% of the sperm will contain the recessive allele. And if the female on the right, again, 50% of the eggs will contain the normal allele, and 50% of the eggs will contain the recessive allele. And so to set up a Punnett square, you set up the potential fertilizations. So if we put the sperm across the top and the eggs down the side, our first potential fertilization is having a sperm with a dominant normal allele fertilize an egg with a dominant normal allele. So the individual will have two dominant alleles. That individual will be homozygous dominant, and that individual will be phenotypically normal. If you cross, if a sperm with a recessive allele fertilizes an egg with a dominant allele, you're going to get a heterozygote with a dominant allele and a recessive allele. But still, that individual will be phenotypically normal because normal is dominant and it's going to mask that recessive allele. And so again, if you have a sperm with a dominant normal allele, an egg with a recessive allele, you get a heterozygous individual that is phenotypically normal. But you can also get a sperm that contains a recessive allele fertilizing an egg that contains a recessive allele. In that case, you get two recessive alleles. That individual is homozygous recessive. And that individual will show the disease, and if the disease is lethal, that individual will die of the disease. And so there is a 25% chance that two heterozygotes will produce a baby that's affected by the genetic disease. Well, what is the probability that two individuals that carry genetic disease will reproduce? It depends on the size of the population. If we look at these king penguins in South Georgia Island near Antarctica, has a very huge population. And typically, genetic diseases are rare in a population. So when you have a huge population size like this, it would be very rare that you would have two individuals that carry the disease reproducing. With inbreeding, negative traits are usually recessive, and negative traits are typically rare in a population, but again, that depends on population size. However, negative traits run in families, and so what happens to the probability of a negative trait being expressed if you make babies with a close relative? Well, this is happening in a small isolated mountain lion population in the Santa Monica Mountains. And so inbreeding becomes a big problem when population sizes get small. And so, excuse me, you have two unrelated mountain lions breeding, P13 and P12, and they produce 3 offspring. But because the population size is so low, it ends up that P12 ended up breeding twice with one of his own daughters, P19, and produced kids, P23 and P24 one time, and then 3 mountain lions, P32, P33, and P34 another time. Okay, and so that's inbreeding. That's when an animal reproduces with someone close, with another animal closely related to it. And you have a much higher probability of having genetic diseases expressed when that happens. If you look at cheetahs in Africa, the cheetah population crashed a long time ago, experienced what's called a bottleneck. And so you have an extremely inbred population of cheetahs. They're so genetically similar that if you take a piece of skin from one cheetah and graft it on another cheetah, there's no tissue recognition. Think about a kidney transplant. If you were going to donate a kidney to me for an A in this class, and I took your kidney, it's highly unlikely that that would be a successful kidney transplant, because my body would recognize your kidney as foreign and would likely reject that kidney. And that's why when they do organ transplants, they want someone closely related to you. Well, these cheetahs are closely related that they don't reject tissues from one another, which indicates a high degree of inbreeding. Well, what are the consequences of that in the cheetah population? You get abnormal sperm. And so you might end up with a sperm cell that has 2 heads, or you might end up with a sperm cell with 2 tails. And abnormal sperm is associated with sterility. Another consequence of inbreeding is low testosterone levels in males. And of course, since negative traits tend to be expressed with inbreeding, you get high juvenile mortality. So inbreeding increases the probability of unfavorable traits being expressed. The negative traits are usually recessive. The negative traits are rare in a population. And since they're recessive, the only time they're expressed is when the individual is homozygous recessive. If the individual is not homozygous recessive, the negative trait will be masked by a dominant allele. And negative traits run in families. And that's the problem with inbreeding, is you have individuals that are closely related, increasing the probability of a homozygous recessive individual that expresses the trait. So in summary, inbreeding tends to fix undesirable traits. You have high juvenile mortality associated with inbreeding. And inbreeding results in the loss of genetic variation, which is often referred to as heterozygosity in a population. Inbreeding reduces a population's chance of surviving environmental change. And so all of this relates to genetic diversity, and we're going to be talking about the size of nature reserves later in the course. And to have genetic diversity, you need a large population. And typically, to have a large population size, you need a large nature reserve. Genetic diversity can be seen at many levels. At the lowest level, if you have 2 different alleles for a particular trait, i.e. the individual is heterozygous, that would be genetic diversity at the gene level. In this class, we're going to be talking about genetic diversity mostly at the population level. We want populations with a lot of genetic diversity. We want populations with a lot of heterozygosity. And so how big a population do you need to have to prevent these problems with inbreeding? Well, it depends on if you're talking about short-term survival, which is 100 years or less, or long-term survival, which would be greater than 100 years. These numbers come from mammal populations. I want you to know the numbers. And so the minimum viable population size, or MVP, the number of individuals you need for short-term survival, 100 years or less, is 50. The minimum viable population size for long-term survival, if you want your population to survive greater than 100 years, you need a population size of at least 500 animals. Some of this data comes from a study that was done on bighorn sheep. You're actually going to be reading the article on this study. And to summarize what the study found, populations of 100 plus bighorn sheep persisted up to about 70 years. If the populations had fewer than 50 animals, they went extinct in less than 50 years. And the author states that genetic deterioration was the primary reason for the decline of the small population. So in summary, genetic variability is extremely important to the long-term survival of wildlife populations. Genetic variability gives the population resistance to disease, and genetic variability gives a population the ability to respond to environmental change. And a buzzword that's associated with genetic variability is hybrid vigor. And so again, you want large population sizes because you want to preserve that genetic variability and you want to prevent inbreeding.